Imagine a virtual cell for a second, just a purely mathematical computational representation of a living cell.
Right.
Back in 2002, the Nobel laureate Sidney Brenner, he proposed this exact concept. He argued that if we could somehow map every single cell in existence, every variation, every tissue, every species into one universal landscape, it would completely change how we understand life.
Yeah, it was a massive, almost philosophical proposition at the time, building a single unified map of life at its most fundamental functional level.
Totally. And for decades, that just sounded like, science fiction. But today, we are jumping into a deep dive on a groundbreaking Nature paper. It's titled, Universal Cell Embedding Provides a Foundation Model for Cell Biology. And honestly, the reality is, Brenner's virtual cell might actually be here.
It really might.
Yeah.
It's kind of staggering.
So today's mission for you listening, we are going to unpack exactly how researchers built something called UCE. That stands for Universal Cell Embedding. It's an AI foundation model that essentially acts as a universal translator for single-cell biology. We're going to look under the hood at the math, the architecture, and what it means when an AI starts organizing 36 million cells completely on its own.
And we should clarify, we are talking about a model that isn't just memorizing data sets here. Because of the really unique way it processes biological info, it can map and accurately understand biology from species it has literally never even been trained on.
Which is mind-blowing. Let's get right into the mechanics of this, because the computational barrier they had to overcome first is, well, it's notorious. We all know how messy single-cell RNA sequencing data gets.
Oh, yeah. Messy is an understatement.
Right. Like, if you sequence a lung cell in one lab and then sequence the exact same type of lung cell in another lab across the country, the resulting data sets often look entirely unrelated computationally.
Right, and that is what we call batch effects. It is the absolute bane of computational biology.
So explain that a bit more. What exactly is the batch effect doing?
Well, think of it like taking a photo of the exact same person, but using two different cameras in different lighting with different lenses. The person is the same, but the pixel data is completely different. In biology, it's the artificial noise from the experimental setup. So the specific chemical reagents the sequencer machine used, even the temperature in the room.
Wow.
Yeah, it completely drowns out the actual biological signal.
So historically, researchers have just been stuck building these isolated, bespoke machine learning models for every single data set just to correct for that localized noise.
Exactly. You couldn't easily query across data sets, which basically means discoveries stay locked in these tiny silos.
Okay, so how does UCE actually strip away those batch effects to create a universal language? I really want to unpack this step by step.
It does it by fundamentally changing the representation of the cell. So UCE treats the cell simply as a bag of RNA.
A bag of RNA.
Right. It doesn't rely on pre-existing genetic network graphs or complex topological rules. It just looks at the transcriptomic output and samples the expressed genes. And it weights that sample by their expression level.
So highly active genes are more likely to be drawn from the bag.
Exactly. The louder the gene, the more likely it gets picked.
Wait, but if it's just grabbing the loudest RNA from the bag, aren't you losing the subtle regulatory networks, like, the lowly expressed transcription factors that are actually pulling the strings?
That is a highly critical trade-off. And, well, we will definitely touch on that limitation later. But for now, the model captures enough of the holistic transcriptomic state to build a very accurate profile. Now, the absolute genius of UCE, the thing that blew my mind, is the next step, the translation phase.
Okay.
Instead of feeding raw RNA sequences or gene names into the model, UCE translates each sampled gene into its corresponding protein product.
Oh, wow. So it projects it directly into protein space.
Yes. It uses this massive 15 billion parameter protein language model called ESM2.
I need to pause you right here because converting the RNA into protein embeddings, that seems like a massive computational detour. Why go through all that effort? Why not just use the gene sequences?
Well, because gene names in raw DNA sequences don't align perfectly across species.
Oh.
Right? So if you try to match homologous genes between, say, a mouse and a human, you get incredibly messy alignments. You lose a ton of data.
Mm-hmm.
But by translating the sequence into the final protein shape, the actual functional machine of the cell, the model becomes completely genome and species agnostic.
That makes so much sense. The AI doesn't need to care about the underlying evolutionary drift of the non-coding DNA. It just reads the universal protein language.
Precisely. It's like spelling a word differently in American versus British English. The letters are different, but the meaning is the same. UCE just looks at the meaning.
It's basically a biological Rosetta Stone. You take incredibly diverse regional dialects, which are the raw data sets from different species, and you translate their root meaning into a universal base language, the ESM2 protein embeddings.
That is exactly what it is doing. And once it had those protein embeddings, it structures the info. It takes a sequence of 1,024 of these translated genes. It sorts them by their genomic location, groups them by chromosome, and then caps the entire sequence with a CLS token.
A CLS token. Wait, that is straight out of natural language processing, right? Like ChatGPT stuff.
It is, yeah. In NLP, a classification token acts as an aggregator for the entire sentence.
Yeah.
So in UCE, by the time that sequence passes through the network, that single CLS token mathematically represents the holistic identity of the entire cell.
That is wild.
Yeah, and this 1,024 gene sentence is then fed into a 33-layer, 650 million parameter transformer.
Okay, and how is it actually trained? Because I know the paper really emphasized that it wasn't given human labels.
Right, no cheat sheets. It relies entirely on self-supervision. So during training, they mask, or basically hide, 20% of the expressed genes in that sequence.
Like a fill-in-the-blank test.
Exactly like that. The model is asked a simple binary question. Was this specific gene expressed in this cell? It guesses, checks the actual data, updates its weights, and tries again. Millions and millions of times.
So by filling in the blanks, it has to understand the deep context of the whole cell. And to accurately guess the missing genes, it naturally learns this rich, batch effect-free representation. Like, it physically has to ignore the lab-specific noise just to consistently guess the biological truth.
You nailed it. And this translation and masking mechanism is what allowed the researchers to scale up to an unprecedented level.
Okay, let's do a quick recap for you listening before we move to the really massive scale of this. So batch effects ruin data integration, right? UCE solves this by treating cells as a bag of RNA, grabbing the loudest genes, translating them into protein embeddings using ESM2, and then using a transformer to predict hidden genes.
Perfectly summarized.
So here's a question for you to chew on. If you were to build a sentence out of your own current biological state right now, what genes do you think would be yelling the loudest? Something to think about as we move to phase 2.
Because phase 2 is where it gets crazy. The researchers use this mechanism to create the Integrated Megascale Atlas, or the IMA.
Yes, the IMA. They fed the UCE model 36 million cells.
36 million, spanning over 1,000 uniquely named cell types across 50 tissues and 8 different species.
And again, all processed without a single human label telling the model what those cells were. It just figured it out.
And this is where the emergent behavior of the foundation model really reveals itself. Despite having no labels, the math organically organized the cells perfectly.
Let's talk about the macrophages, because that example really stuck out to me in the paper.
Oh, it's a great example. So in older computational models, a macrophage from the brain and a macrophage from the liver, they would cluster far apart on a graph. The tissue-specific expression and the batch effects just completely mask their shared identity.
But in the UCE space...
In the UCE space, macrophage clusters from 73 different tissues align beautifully together. In fact, 72% of tissue-specific macrophage clusters embedded closest to a macrophage cluster from another tissue entirely.
Wow. So the model mathematically saw past the tissue-specific disguise, basically, and recognized the core macrophage architecture underneath.
Exactly. It stripped away the noise.
That is so powerful. But the paper also noted it uncovered developmental lineages, which I honestly find even more fascinating.
It's stunning. So you have the 3 primary embryonic germ layers, right? Mesoderm, endoderm, and ectoderm. Without giving the AI any programming about embryology, UCE grouped mature, fully differentiated cells by their germ layer origin with over 80% accuracy.
Wait, really? It can look at a mature cell's current transcriptomic state, read that protein sentence, and mathematically deduce its ancestral origin.
Yes, because those ancient developmental pathways, they still leave an echo in the cell. And UCE is sensitive enough to pick up that transcriptomic echo.
That is, I mean, that's just incredible.
And it gets better. It maps directly to our established biological frameworks. The distance between cells in the UCE coordinate space perfectly matches their biological distance in the official cell ontology tree.
Meaning what? Exactly.
Meaning if 2 cells are close cousins on the human-made biology tree, they are mathematically close together in the AI's virtual space. And this holds up beautifully up to 5 hops apart on the ontology graph.
It's literally the Waddington landscape rendered in high-dimensional math.
Yes.
For those who might not know, biologists have used that Waddington metaphor for decades. The idea of a marble rolling down a hill into diverging valleys to explain stem cell differentiation. UCE actually built the quantitative coordinate system for that hill. It's proving that it isn't just memorizing clusters, right? It's learning the underlying physics of developmental biology.
Exactly. Which brings us to the ultimate test of any foundation model, zero-shot generalization.
Zero shot. Let's do another quick recap for the listeners here before we define that. So UCE built a 36 million cell atlas, clustered cells perfectly without human labels, and identified cross-tissue similarities and developmental lineages purely on its own.
Right.
So, question for you listening. How does it change your view of biology to know that a machine can organically deduce the developmental history of a cell just by reading its current protein expressions? It's pretty wild.
It really makes you see cells differently. But yeah, zero-shot, organizing 36 million cells you've trained on is a massive achievement. But how does the model handle data it has literally never encountered?
Right. Taking the final exam completely cold, no fine-tuning, no weight updates. What is zero shot for the uninitiated?
Zero shot just means you take a completely new data set and you ask the model to map it into its universal space without retraining the model at all. So they tested this on Tabula sapiens version 2. It's a massive, unseen data set of human cells. But crucially, it was sequenced using both droplet-based and plate-based technologies.
And that hardware difference usually causes catastrophic batch effects, right?
Usually, yes. It splits the data apart.
And UCE mapped it.
Completely seamlessly. It embedded the new data directly into the universal space, correcting the droplet versus plate batch effects right on the fly. It's significantly outperformed other models like Geniformer and SCGPT. Actually, it beat Geniformer by nearly 14% in overall score and over 16% in biological conservation.
But Tabula sapiens is still human data. We've seen a lot of human data. Here's where it gets really interesting. What happens when you feed it an entirely new species?
This is where UCE truly flexes the power of that ESM2 protein translation we talked about earlier. So the training data included humans, mice, pigs, zebrafish, macaques, 8 species total, but 0 birds. Avian DNA was completely absent from the training data.
None at all.
None. Yet researchers fed it a data set from a developing chick heart and a chick retina.
And it was able to map the chicken cells to their mammalian counterparts.
Flawlessly. I'm not kidding. It mapped chick eye neurons directly to mouse lemur neurons in the mathematical space. It realized they were biologically equivalent despite being separated by literally hundreds of millions of years of evolution.
Wow. Because, again, it doesn't care about the bird DNA sequence. It sees the functional protein, recognizes the shape and function, and says, yep, this is an eye neuron. That shared evolutionary history is just baked into the protein language.
Exactly. And it gets even more interesting when it comes to human error.
Oh, the green monkey data set. I love this part.
Yes. So in a data set from a green monkey lymph node, there was a population of cells originally annotated by the human researchers as B cells. But when they were mapped into the UCE space, these supposed B cells formed a distinct cluster far away from the other B cells. They actually landed right next to the T cells.
So the AI explicitly disagreed with the human annotation. It flagged an anomaly.
It did. And so the researchers went back, ran a differential expression analysis on that specific cluster, and found that UCE was completely right.
That's amazing. What were the markers?
Well, those cells predominantly expressed canonical T cell markers. Things like CD3, AB1, IL-7R, HCS, and GZMNK. The human annotation in the original dataset was just wrong.
So UCE organically corrected a human error in a species it had never even been trained on.
Right. It operates as a mathematical truth checker for biology.
Okay, mini recap time. Zero-shot learning requires no model retraining. UCE conquered new sequencing tech, mapped unseen species like the chicken to known mammalian biology, and even corrected human annotation errors in a green monkey.
Yep, it's a very robust system.
So, audience, think about this. If a model can identify the biological cousins of a chicken cell in a mouse lemur, what does that tell you about the deep shared evolutionary history of all life on Earth? It's all connected.
It really is.
But let's take this a step further. It's one thing to organize data and fix typos in existing annotations. It is another thing entirely to generate entirely new biological hypotheses. I really want to dig into the hunt for the Norn cell because this is where the paper just blew me away.
The Norn cell discovery it perfectly illustrates why universal embedding space is so powerful. So Norn cells are these highly elusive, recently discovered fibroblast-like cells found in the kidney. Their defining characteristic is that they produce erythropoietin, or EPO. That's the hormone that stimulates red blood cell production.
Okay, so if you're a biologist looking for a Norn cell, you're usually just scanning data sets looking for that one EPO gene to be turned on.
Exactly. That's the standard marker gene approach. But the researchers wanted to see if UCE could find Norn cells without relying on just that one key word. So they took an existing data set of mouse renal cells, mapped it zero-shot into UCE, and identified the specific cluster that corresponded to Norn cells.
Okay.
Then they trained a very simple logistic classifier to recognize the mathematical signature of that specific cluster in the UCE space.
I see. So they basically isolated the mathematical scent of a Norn cell.
Exactly. And then they unleashed that classifier on the entire integrated megascale atlas, all 36 million cells. They essentially asked the model, where else in the known biological universe does this specific mathematical signature exist?
Wow, an unbiased universal search engine. And it found them in the heart and the lungs, right?
It did.
But hang on, I read the source material closely on this, and there's a glaring paradox here. The source notes that these newly found cells in the heart and lung, they expressed canonical Norn markers genes like DCN, LPAR1, COL1A1, CXCL12, CSH, but they often did not express EPO.
Right.
If Norn cells are literally defined by producing EPO, how can the AI claim it found them if the EPO gene is quiet? That just sounds like a false positive.
Right, and that right there is known as the EPO paradox. And it perfectly exposes the major flaw in how human researchers typically hunt for rare cells. We rely heavily on single marker genes, but EPO transcription is notoriously bursty. Yeah, the gene turns on briefly, mates the transcript, and then the mRNA degrades incredibly fast. The overall expression over time is actually very low. So if you are a researcher only looking for the EPO transcript, you will completely miss these cells if you happen to sequence them between bursts.
Because we are looking for one specific leaf on the tree, and if it happens to have fallen off, we assume the tree just isn't there at all.
Precisely. But UCE isn't looking for one marker gene. It is analyzing the holistic, universal state of the cell. That entire 1,024-word protein sentence. UCE recognized the fundamental baseline identity of the Norn cell architecture in the heart and lungs, even when the EPO gene was temporarily quiet.
That is wow.
It shifts us from searching by keyword to searching by deep contextual meaning. It discovers connections that traditional marker gene hunting completely misses.
Okay, final mini recap before we hit the limitations. UCE can be used as an unbiased search engine across all biology. Researchers used it to hunt for kidney Norn cells, found them in the heart and lung, and UCE succeeded where traditional marker gene hunting fails because it looks at the whole cell state, not just one bursty gene.
Spot on.
So listener, what other rare cell types might be hiding in plain sight in our organs, just waiting for a universal translator to spot them? It implies there are massive blind spots in our current cellular atlases just because we've been using the wrong search tools.
Oh, absolutely.
But before we declare AI the undisputed king of cell biology, we do need a reality check. We need to talk about the limitations. Where does UCE fall short right now?
Well, the paper is very transparent about several key caveats. First and foremost, it is a black box.
Classic AI problem.
Right. Like many large foundation models, we know it works, but interpreting exactly why certain embeddings arise is incredibly difficult. We really need better interpretability tools to map its mathematical decisions back to mechanistic biological pathways.
It's like having a genius lab partner who gives you the correct answers every time, but flat out refuses to show their work.
Exactly. It's frustrating. Second is the bias in the training data. While 36 million cells is massive, it is heavily skewed toward mammals, specifically humans and mice. And it's heavily biased toward brain tissue, simply because that is what researchers have historically sequenced the most.
So it needs more diverse data.
Much more. The model needs vastly more diverse data to ensure it generalizes perfectly across the entire tree of life.
And what about that 1,024 gene limit you mentioned earlier? Because a typical cell expresses thousands of genes simultaneously. Doesn't capping the sentence at 124 tokens mean we're losing some resolution?
It absolutely does. That is the third major limitation. Because it samples a set number of genes, some fine-grained, highly specific quantitative variation is lost. So it is excellent at mapping the broad architecture and cell type, but it might miss incredibly subtle state changes or, transient regulatory signals.
Makes sense. And I imagine it doesn't entirely replace traditional batch correction for every scenario, right?
Right. That is the fourth limitation. While it is incredible at zero-shot alignment across massive data sets, if you are a researcher dealing with known, highly specific experimental confounders, In a small, localized dataset, traditional batch correction methods might still offer more granular control.
So putting all of this together, we have a model that translates the chaos of RNA sequencing into a universal protein language. It organizes millions of cells without human intervention, maps evolutionary homology across unseen species, corrects human annotation errors, and can uncover hidden, rare cells that traditional methods miss. Where does this actually leave us in the grand scheme of biology?
If we connect this to the bigger picture, it really leaves us standing on the threshold of Sidney Brenner's vision. He argued that cells are the real units of function and structure in biology, and we needed models that are computationally efficient, predictive, and capable of identifying entirely new biology.
And UCE does that.
UCE provides the foundational scaffolding for that virtual cell. Despite its flaws, it proves that there is a computable universal language underlying all cellular life.
It really is the ultimate biological translator. So to wrap this up, thank you to everyone listening for joining this deep dive into the microscopic AI-driven universe of UCE.
It's been a fun one.
It has. And I want to leave you with a final provocative thought. If an AI model can find a kidney cell's long-lost cousin hiding in your lungs based purely on the mathematical similarity of their protein languages, what other hidden biological networks are governing human health and disease that we simply haven't had the vocabulary to see yet? Keep questioning, keep learning, and we'll catch you on the next deep dive.